Dr. Manish Manoria, sir, Executive Director, Truba Group. Dr. Suren Jain, sir, Director, Truba Institute of Pharmacy. Dr. Rajesh Singh Pawar, sir, Principal, Truba Institute of Pharmacy, and all the participants in this web conclave. We are very happy to share with you. This is our 11th web conclave online lecture series. This web conclave is organized by Truba Institute of Pharmacy in association with Bhopal local chapter, Society of Ethnopharmacology, India. This 11th web conclave is on the topic of molecular docking, dangers, utility, challenges, and application. By this lecture will be delivered by our eminent speaker, Professor Dr. Robert Dodson. This Truba group of institutions, a pioneer institute providing quality education in pharmaceutical sciences, engineering and technology, commerce. This is situated in Bhopal, Madhya Pradesh, India. Truba is known for providing the skill-oriented education and imparting world-class infrastructure. Truba is established in year 2001, providing the quality education. Truba Institute of Pharmacy started in 2004. Now, may I request Dr. Rajesh Singh Pawar, sir, Principal, Truba Institute of Pharmacy, to address with the welcome note. Over to you, Pawar, sir.
Thank you, Professor Dhote. In this evening, dear participants, my colleagues, Team Truba, and guest of the day, Dipti ma'am, our BOG member, Truba Group. Good evening to all of you, and good morning at US. I, Rajesh Singh Pawar, welcome to all of you in this 11th Interdisciplinary ETHNO-SCIENCE Web Conclave lecture series. On this occasion, today is with us Dr. Robert, who is eminent speaker, professor in biomolecular science, School of Pharmacy, University of Mississippi, USA. And on this occasion, I would like to welcome our colleague, Dr. Suren Kumar Jain sir, and our executive doctor, Dr. Manoria. So welcome to in this event. Now I would request to Professor Dhote, he is requesting to Dr. Robert to start their talk. And before going to start the talk, I would request to Dr. Manoria to say a few words to our participants. Dr. Manoria. Manoria, sir. Manoria, sir, please. I would request to Suren sir to say a few words to our participants.
Thank you, Rajesh sir, for conducting such a nice interdisciplinary conclave in association with the Bhopal chapter of Ethnopharmacology. On behalf of Truba Group of Institute and Truba Institute of Pharmacy, I would like to welcome Dr. Robert J. Dodson sir for giving his consent to deliver a nice talk on molecular docking, dangers, utility, challenges, and applications. Might be, as I am also a person from medicinal chemistry, I did my post-graduation and PhD in medicinal chemistry only. So might be the attendees who were attending this conclave will get a full fruit knowledge of molecular docking and molecular modeling. So I, from my side and from all the Truba Group of Institute person side, I welcome you, Dr. Robert, for delivering this particular talk. Thank you, sir. Thank you very much.
Thank you. Dr. Robert, you may proceed.
All right, thanks very much. Please let me know approximately when I should stop for any questions.
Okay.
what time would be a good ending point? Shall I go until about 7:20 your time? Can you hear me clearly?
Yeah.
Robert Docking from Department of Biomolecular Sciences, School of Pharmacy, University of Mississippi, USA. Sir have done PhD from University of New York, Canada. He has done postdoc from University of California, USA. Currently, sir is working as Associate Dean of the Graduate School, University of Mississippi, USA, Professor of Medicinal Chemistry, Department of Biomolecular Sciences, School of Pharmacy, Mississippi, USA, Director of US National Institute of Health, NIH. Sir have currently 2 NIH grants for glycosines and tuberculosis. Sir have more than 90 peer-reviewed publications, 60 invited research seminars, 60 conference talks, more than 180 conference poster presentation, completed 40 funded research projects from various funding agencies. Now, may I request respected Dr. Robert Docking, sir, for today's talk on molecular docking, dangers, utility, challenges, and applications. Over to you, Robert, sir.
Thanks very much. Can you hear me clearly now?
Yes, sir.
Great. Thanks very much to everybody for joining this presentation. Thank you to everybody for the invitation, particularly to Professor Pawar, Professor and Principal at Truba Institute of Pharmacy, for contacting me personally. I really appreciate it. And it's great to have the chance to speak to you today and to try to inspire you. One of the themes I feel that's really important in doing research is to try to do it well. So that's sort of a background theme to my presentation. Thanks as well to the Society of Ethnopharmacology. That's one of the major backgrounds to the research that we do here. So I'll mention a little bit about that as we go along. I wanted to thank at the beginning my research group and my sources of funding, because without them, the research just wouldn't get done. So let me point out to you, first of all, on the left, my senior PhD student, Iolua Adara-Bigby. She's about to complete her PhD this summer. And it's been a thrill over the years to have a number of PhD students working with me, and I'll show you a couple of more of them on the next page as well. And then I have a current PhD student who started last year, Priyanka Samanta. And you can see for the students, I've listed some of their degrees, particularly those who came from India. Then I have 2 research scientists working with me. One is Dr. Pankaj Pandey. He is actually from Madhya Pradesh. You can see his master's degree listed here. And he came to do his PhD with me. He's currently working as a scientist in our National Center for Natural Products Research and continuing to work with me as well in collaborations. And then I have Dr. Sushil Mishra over on the right here, who is working with our glycoscience research. I also like to work with undergraduate students. You can see my current 3 undergraduate students working with me, Ashton, Zainab, and then Lara will be starting later this month for the summer. Also, I'll be mentioning some research contributions from 3 of my prior PhD students, Dr. Prasanna Sivapakasam, who's a scientist at Bristol-Myers Squibb, Dr. Gang Fu, who's a scientist and computer scientist now working at Amazon Web Services, and Dr. Khaled Al-Okali, who's a professor appointed at Tanta University in Egypt and also has been working at Temple University. My funding has really been very helpful. Apart from the NIH grants, which were mentioned, I've also been very much appreciative of funding from the National Science Foundation of the United States. When you come to visit our University of Mississippi, and I hope you will, one of the most famous scenes is shown here on the left. where you can see how crowded it can be at times when there is no COVID-19 pandemic, when people come to celebrate our American football games, which are held approximately just only about 8 weekends per year, but they're really a popular highlight. And let me mention before I move on, you know, my sorrow in hearing about the troubles of coronavirus 19 in India as well as in other parts of the world. We've had terrible trouble, and now, of course, people like to do social distancing, and this type of celebration wouldn't be allowed. But just right next to that famous park we call the Grove on campus, looking right over it is the School of Pharmacy, which you can see right here. This is our major research center where my students and scientists have their office, and where we've been conducting research in our School of Pharmacy for about 110 years at the University of Mississippi, which was founded in 1848. We've had for more than 50 years an ongoing project about marijuana or cannabis. You can see it growing here in a field right on our university campus, the only place authorized for many years to grow marijuana in the United States of America. It was grown for research purposes in order to be able to send supplies to any lab in the nation who wanted to do research about the interesting properties of this plant. And similarly, over the years, we've had a traditional medicinal plant garden at the University of Mississippi, established in the 1960s, where we've been able to do all sorts of studies related to ethnopharmacology, as well as just trying to find useful active ingredients from plant species, which could help for human health or for agriculture. I wanted to mention as well, in my role in the graduate school at the University of Mississippi, I welcome your students, those of you who are currently doing your bachelor's degree or master's degree. You may consider applying to University of Mississippi. We have 2,300 students currently doing various master's and doctoral degrees, as many as 140 different degree programs. So I encourage you to consider, you can fill out an application or interest form and find out more, and feel free to contact me as well. And then I wanted to mention about the research center that I work on now. We have a 5-year funding to focus about glycoscience, and there you can see my research scientist, Dr. Mishra, hard at work at our computational facilities. And we're available to assist others in research. We've been holding workshops. Dr. Pawar was able to attend one of ours recently, and we will have a one-day conference coming up in the start of July. You can see it listed on my webpage. It's free to all in the world to attend, so I encourage you to attend. You can present about your research there, and also learn from the great guest speakers that we have coming. So let me talk about docking. We have, I will of course explain what I mean by docking and give a brief history of the docking. Then I'll show you some comparative docking studies, because I feel that in these comparison studies, we can start to get a sense of what method is accurate for the docking that we will do, and to see some of the dangers of doing docking in the wrong way. And I'll show you some examples of docking from our work, which I think my research overall focuses on computational medicinal chemistry. So when we consider about the word docking, then of course it might be good to just do a Google search and see what is docking. And the #1 hit you'll find if you look for docking is the docking of dog tails. So when I heard about that, I was thinking, okay, what exactly do we mean by docking of a dog tail? Is this what we mean here? A dog's tail fitting into the mouth of the dog? Well, no, the #1 meaning of docking is actually to do with removing the tail of the dog, such as seen on this dog here, whose tail has been cut supposedly for visual appeal. I'm not sure how happy he was to have his tail cut off, but that's not what we're talking about. We're really talking about more of this kind of docking, fitting one thing into something else. So we consider instead the analogy to the boat. When a boat is well parked, then we say it is docked. So we're thinking about that. We're thinking about finding an optimal location where we see, and here it's sort of obvious, that large container ship should be going into that dock area right here. Some other times these container ships can run into trouble, such as what recently happened in the Suez, where it's just not being docked properly. It wasn't even supposed to be docked in the middle of this canal. So we're thinking about docking, but instead with molecules, and that means taking a small molecule like the one found over on the left here, and seeing where is there any place on this protein, which is shown on the right in purple, where would the optimal place for this molecule to fit in to have a useful pharmacological effect. So I think of molecular docking as fitting one molecular species to another. We could think of it in terms of a surface representation of a protein as shown here. And we might consider, look, here's a hole in the protein. Is that a possible place? You could try and take this molecule and fit it in various places on the surface of the protein to see where it interacts. And we should think about this as basically, with those of you who are familiar with the chemistry concept of the covalent bond, docking means interactions without covalent bonds, primarily with non-bonded forces. Those non-bonded forces are, you might call electrostatic forces, the interactions that cause attraction between slightly positive functional groups of molecular species and slightly negative. Those groups will be attracted to each other. And also then there's a hydrophobic effect where hydrophobic portions of molecules, those that are repelled, that repel water and are repelled by water, they like to be associated with each other. The other major factor for docking is just about the volume or the size of molecular structures and how they can fit into each other from that point of view. So I always like to use docking example like this. Here's the place for docking and here's the object being docked in. Okay, so let me mention that all of this work fits under the area that we call computer-aided molecular design or molecular modeling. When we consider about that, then we think of that as employing computers to try to help in design, discovery, and optimization of bioactive molecules as putative drugs. And another way of looking at it is to say we're just trying to understand more about the properties of molecules and how they interact with each other. And so part of that is actually even just making cool pictures so that we could have a vision for how things interact to help excite scientists' minds. If you search for the history of docking, the very first paper that was published that has docking in its title or information is actually from the 1960s regarding space vehicles and how you can get them to be positioned close to each other. And then there was a scientist whose last name was Docking, and that's of course irrelevant. If we move forward to the 1970s, there we can see the third list on here, the first reference to docking of molecular structures from 1979. You can see that docking maneuver at a drug receptor. And then after that, things have exploded so much that if you look now for the similar search term of docking, you'll find, for example, I checked in Scopus recently, 100,000 different references to docking and about 80,000 to do with molecular docking. So it's a very common method now in drug discovery. The history of docking in terms of organized scientific programs, computer programs, would be going back to the University of California, San Francisco, where they developed a program simply called DOC. And so you can see that currently the website looks like this. It has a new release of 2 versions of the DOC software, one 3.7 and another current release of 6.9. 2 different research groups have, the Kunz and Soche group have developed this simultaneously. You can see other groups involved, such as the CASE group. And so it's important to look at that type of docking software just to understand more about the way that the research is done. So this was developed back in the 1980s, and it continues to be developed actually in different computer languages. So this development of software can make it convenient for other users to use the software, but then it opens up the question, what is the right use, what's the correct use, and which software to use? The original docking approach involved, instead of representing all the details of the molecular structure, it involved trying to make a representation of the space where a molecule could dock into a protein. So here at the right, you can see a purple protein representation, and those yellow spheres represent space where there's room for something to dock. If there's just a single sphere by itself, it's not big enough for a large molecule to fit in. Now at the upper right, you can see a bunch of spheres representing a so-called small molecule drug, or we might call it a ligand. In this type of docking, we tend to say protein ligand docking, where ligand refers to a small molecule which will be not covalently bound to the protein. And so we could also, as was done in the original dock software, represent the ligand by a series of spheres. Then it's just a matter of seeing how things match. And they developed an algorithm involving looking for pairs of ligand spheres which could match with space, or with basically what we say receptor sphere pairs, and considering all these different matches, and then gradually being able to fit the smaller molecule into the larger molecule. Now, what often happens is that you end up with several different possible places where the molecule can dock into the biomolecule. So those are called docking sites. And it's important then to have a score system to be able to figure out which site is optimal. There are many, many different types of software available now for docking. One of the most famous ones was developed at Scripps Institute, also in California, the AutoDock software. Currently, you can see they have AutoDock 4. There are a wide variety of detailed numbers, such as 4.6.3, and then AutoDock Vina. They also have other AutoDocks under continuous development these days. From the Cambridge Crystallographic Data Center, they developed the GOLD software. A company formerly known as Tripos developed Sybil and the FlexX software, which I'll mention briefly. And then Biovia, a company which used to be known as Acceleris, developed various types of docking software, including CDocker. One of the main ones that my research group and our School of Pharmacy has been relying on for about 15 years now is the Schrödinger software. It's commercial software. It's a bit expensive, but they do have discounts available for institutions in India, I know. And so we quite often make use of the Glide, which is just their docking software algorithm. And I'll mention some results from the Schrödinger software later in today's presentation. Then a Milagro Virtual Docker is another example. So with all of these programs, when you use the software, you should expect different results compared to if you use a different program. Each program gives different results. That makes it very confusing to know which is the best way to proceed in research. It's very challenging. And the factors to consider are about when the program was set up, how big was the database of test compounds that were used in the development of the software, as well as in the testing and validation. So one of the things that I like about the Schrödinger software is that it did have a very large validation set when it was originally set up, and they continue to do various developments to their software. At the heart of it, we won't talk a lot about the mathematics today, but we need to have a way for to represent the interaction between two molecular species that are not chemically bound. So that's just like I was describing before. You have two objects which are polar opposites to it, one that's positively charged and one that's negatively charged. And so they will tend to be attracted to each other, but they have a physical shape, so they can't actually overlap with each other. That would be too close for comfort. So to do that type of model, you can have a model like this where there's an attractive term with a positive sign plus A, and then a negative term. B is also positive, but because of this minus sign, that shows that there's a negative term. A balance between positive and the negative will mean that there's a certain optimal distance between these two objects. R, small r is the distance between the objects. And so you end up with a curve which illustrates that there would be a minimum or an optimal distance between the two objects. You can see over here specifically electrostatic term involving the charges. This other term here is what we call the van der Waals term. It has to do more with the volume and the overlap of the structures. But any object, any molecular object, whether it's actually charged overall like an ion or whether it's a neutral species, it will have this type of van der Waals interaction, which is important for long-range attraction and short-range repulsion. And then a wide variety of other terms have been introduced over the years. So the challenge partly is having a mathematical function which represents the physics of the way the molecules interact. Then we have also the possibility of including particular values for these letters like A, sub I, and J to do with particular interacting species. So you could say if you have a carbon, which is sp2 hybridized, it will be different from a carbon that's sp3 hybridized. It'll be different from an oxygen. And so you can have these parameters be specific for particular kinds of atomic groups, particular types of molecular environments in order to have a force field and a representation of the molecular structure which is accurate. There are a wide variety of softwares available also for protein-protein docking, and here are some examples. So, the whole development of software in the industry and in the world in general is partly having general software, which is applicable for many different situations, and specific software, which is actually good for particular projects of interest. And so that's part of the challenge that one faces in doing research in this area. Also, there's a chance to just submit your molecule of interest to have a calculation done about docking on the internet itself without even setting up any software on your own computer. This is a very convenient thing, very nice, good for people who have low computational resources at their home institution. But remember that it will be just superficially helpful. Oftentimes there will be errors and mistakes in the results that you get, and the servers themselves mention about that. Don't rely too much on this, they say. So what happens though is that you'll find a wide variety of papers published, even in very high quality, prestigious journals that include what I call bad or lousy docking. I was looking at a paper just yesterday at one of the Nature journals, Nature Communications, and the collection of computational methods that were used in that portion, because most of the paper was about some amazing experimental results, but the computational work was highly dubious in the approach that was taken, including the use of an online server for the docking. So we'll do some examples of comparative docking studies from my own research groups, previous work. Cycloguanyl derivatives docked to DHFR, CSAR, benchmark exercise, and then Ensemble docking. Search for GSK3 beta inhibitors. So for the first example, I'll just show you one-page summary. This is from my first PhD student, Prasanna. So he looked at a very small test case. We were interested in finding new drugs for for curing and treating malaria. So DHFR is a reductase that's a very common target for anti-malarial research. And so we can find some experimental activity for these compounds in terms of how well they're able to inhibit the functioning of this complex protein DHFR-TS. So Prasanna used, together with our summer student, Veritaso, used a variety of docking software, FlexX, Gold, Glide, and Vallegro, and different scoring functions within those. So on the y-axis, you can see the docking score. And notice that in some software, the more negative is considered the better score, like at the top here. And then in others, the more positive is considered the better score, like in the bottom for the Gold fitness score. Now, with the experimental activity expressed as on a log score, 10 to the minus 9 would convert to a 9 on this, this type of scale. So the best activity is over on the right. This type of correlation plot, you can then plot the R squared, and R squared of close to 0 means that there's no correlation, whereas an R squared close to 1 means a very good correlation. So the result is very good for the Gold fitness score, but it's extremely bad for this FlexX C score. And we found also for a variety of other scores that the results were just very poor. So the conclusion is that there is wide variability in the performance. If you don't know the experimental activity in advance, you're trying to do some predictive work, then you're in trouble because you don't know which software will be best. So what can you do? Well, you have to look at systematic trends. You have to look at a group of related molecules and try to see which computational method works best for that particular protein and that particular group of molecules of interest that are similar to the ones that you will be working with. Periodically, there are competitions held to try and improve the methods of docking to find out which are the best. So one of those has been run by the Community Structure Activity Resource, where they have a lot of docking data available. And so we participated in the 2012 benchmark exercise and ended up with a good publication about it, which appeared on the cover of Journal of Chemical Information and Modeling. So this is work of my PhD student, Khaled. So he used a wide variety of methods for docking for 4 different protein cases. And this slide is very helpful because it'll show you some of the choices that can be made when you're doing the actual docking itself in your research projects or when you're reading about it in considering the value of published work. So one thing is up on the upper left, the idea of protein sampling. How do we know what is the correct 3-dimensional structure of a protein, of all the atomic centers that are found in a protein? Well, sometimes we can find the information from experiments such as X-ray crystallography. And so if you have that information, what can you do with it? Do you want to just use it verbatim as it is, or do you need to do further computations on it? So one possibility is to have the structure just rigid as it was found. Another possibility is to allow slight flexibility using methods such as the soft receptor method. We could have induced fit docking where just close to the docking pocket, the protein side chains are allowed to be flexible. Or we could have many different side chains flexible all over. So that's just one dimension of different possible choices that can be made. Another question is, for the small molecules that we will be docking, how do we know what is the correct 3-dimensional conformation for those? Is it better to have a separate piece of software to generate the structures or to generate them within the docking software itself? So you can see here also mentioned in this flow chart different approaches such as pre-generation of conformations or generating them within the software. And so then these orange labels represent a wide variety of different methods that were produced. Once we have a docking pose, how many docking poses do we want to obtain? We could obtain many or few. once we have a docking pose, we have to score it. How can we score it? There are a wide variety of scoring functions. There are different algorithms, such as the so-called ant-based search algorithm, to try and find out all the different possible algorithms, just like the way that a swarm of ants will try and find the optimal source of food. So then some of these models, you can see this is a slightly different view of a receptor where all the side chains have a lot of flexibility. We could simultaneously consider basically thousands or millions of different conformations of the protein representing all these different ones, or we can have just a slight flexibility around certain portions of the protein structure, or we can simultaneously study a variety of X-ray crystal structures in the method known as ensemble docking. So a variety of approaches have been proposed and used. So what was the result? Here you can see on these lines here, CHK1, ERK2, those are different protein targets that were studied in this exercise, in this competition. And then the columns represent different approaches for docking, such as having a rigid protein or soft protein, induced fit docking, ensemble docking. And then here's the percent accuracy in terms of how well the results turned out, because in the end of the competition, the results were shown for exactly where the molecules bind and how well they bind. And so a number of 100% would be good. You can see there are very few results even better than 80%. In fact, the only really good protein case was the LPXC case, where 5 out of the 6 models presented had approximately 70 or 80% accuracy. And so there are a great range of accuracies. Many of the methods failed, even though the authors who prepared the methods were sincere in their development of a method that they thought would be helpful. A method may work well for one protein, but not for another, such as this case here, down to 20% for some of the other proteins. For some proteins, all the methods failed, like for CHK1. Now let's go on to another example, the ensemble docking approach. Gong Fu, my former PhD student, was the lead on this work. He was studying about the glycogen synthase kinase protein, a very interesting protein which has docking pockets in this region between these two lobes. So in his work, he took the advantage of many different X-ray crystal structures represented by their protein data bank label in the first column on the left. And some of them have no small molecule bound to them at all, whereas others have a particular small molecule in their active site. So what we can do is to pull that small molecule out and then use the docking software to fit it back in. And if it fits in optimally, the root mean square deviation of the docked molecule compared to the co-crystallized molecule will be low, close to 0. And that'll mean it's a good docking method. But another method is to say, let's take staurosporin for 1Q3D and dock it into 1Q3W, where Ulster pollen was found, and see if it docks well there. If it docks well there, then with a low RMSD, that means that must be a very good docking method. So that's what we did in the study. So here the red background indicates poor docking, where this is just for self-docking, where you take the molecule like staurosporin from the crystal structure and then place it back in again using the docking software. If it does a good job, the numbers will be low, less than 1.5 angstroms, or 10 to the minus 10 meters. Whereas if the numbers are high, then that means it's a bad method. So you can see here that the best method was glide XP docking and glide SP docking, whereas some of these other softwares did a bad job. Now, if you look at some examples here, this shows some of the variety of how the small molecules were able to dock, which residues were key, and that's the sort of thing that we hope to study from our docking approach. Now, what about for cross-docking? this shows that it was a very big challenge to do the cross-docking, where you take a ligand, say, from one of the protein structures like 1UV5, and see if you dock it into the other proteins, how it does. The other crystal structures have exactly the same GSK3 beta. So this again illustrates the challenges of docking. Most of these backgrounds are red, indicating poor docking in this case. We found once again that the best method was the Glide SP in terms of computational time and efficiency and accuracy of results. So that's the method we did for this work on this slide. So then we were able to use that method, the optimal method, for taking a large database of structures and fitting them into our protein models, not just to one, but in this case for ensemble docking, fitting them into a variety of crystal structures and then seeing which were best. And we used a ligand decoy set to show molecules we expect would not dock well. So then we can get a type of study of here are some molecules which should dock well, some that should not dock well, and to study about the enrichment of the database. In other words, if you were to dock just 10% of the database, would you find all the best molecules, the ones that are true dockers for this particular protein target? They would be selected just in the very small percent of the database which is screened. So the actives that are recovered is plotted on the y-axis. And the best chart here is, after a small amount here, is this red curve. The ideal, the best you could hope for, is this black line. And so the red curve comes closest to that. And that's what we call ensemble docking, where you make use of all of the structures in your docking, not just using a single protein crystal structure. So now let's move forward and see a few summary points. We were able to use the docking algorithm to discover some new ligands which could potentially develop as drugs. So the lessons on docking that I like to summarize are really all presented in the review article from Khalid, which now has been cited well over 100 times. One is to try to pre-generate the ligand conformations. Don't just trust the docking software for that. Don't minimize the protein ligand complex. If you have experimental information about its structure, try and keep the structure as close to that as possible. The role of water molecules in the crystal structure are extremely important, so make sure to analyze their importance and include them if necessary. If you have a lot of experimental information about a protein target of interest, then make sure to use it by using, for example, a hybrid approach with a number of different rigid receptors, in other words, different models of the same protein, or use them combined in an ensemble docking approach with allowing us flexibility around the docking region. And then if you have the case that's often so that you're dealing with a protein for which you do not have any experimental information, then it would be useful to try to make use of any information you have available regarding, is there supposed to be flexibility of the protein? Is it able to dock a variety of molecules of different structure and size? And then try to use induced fit docking as well as flexible side chain approaches. Now I'm going to give a couple of examples, but let me ask the organizers once again, at what point should I stop? Do I have just 1 more minute, or 5 more minutes, or 10 more minutes?
Hello.
Hi, how many more minutes should I talk for?
Yeah. Why not? Why not? You can carry it. Yeah.
For how long?
You have a 10 minutes.
Okay, 10 minutes. Thank you. Okay. Sure, thank you. Okay, so I'll give you a few examples of docking and computational medicinal chemistry projects from my research group at our School of Pharmacy. Here you can see, I'll mention about some protein kinase work we've done. the docking into proteins to find targets of natural products, and then cannabinoid receptor docking. So actually, I mentioned the protein kinase part earlier with regard to GSK3, plus we worked on some other proteins, which are protein kinases that are relevant to Alzheimer's disease. But one of the interesting projects I've worked on together with Mark Hammond at the Medical University of South Carolina is trying to find plants which are endangered species that may, in another generation or so, not even exist anymore in the world. Can we find any useful molecules that could be developed into drugs from such plants? So we had a very nice paper published about a particular rare plant, which is mentioned here from the mountains of Ecuador. And we found molecules that had two completely different kinds of structures as well as kinds of activities, one with glycosides active against hepatitis C virus, and then other molecules that I'll show on the next page, which were active against a potential target for diabetes. So in this case, the docking involved comparing two structures that were very similar. One you could see has an OH at the end, alcohol, and the other one has the O without an H on it, which is the carbonyl, to see about the difference in docking and how the fact that this has the OH on it is allowing it to interact with this glutamic acid to help improve its binding. So that's the kind of study that we can do. At our university, we've been studying about the cannabis plant for many years. So I wanted to show you some results from our work on looking at some of the rarer molecules found from the cannabis plant. You can see 7 and 8 shown here. Their structures vary. Only one point about this stereocenter here, whether that hydroxyl is going back into the page or coming out into the page. And you can see that we end up with completely different, I would say slightly different or rather different docking poses. And we think that this can help to explain the difference in activity between these two structures, where molecule 7 docked better into the structure of the cannabinoid receptor 1 protein, CB1, and that correlated with the better experimental activity. So we've done a detailed study of many of these molecules, which will be submitted for publication soon. And then here's some docking showing the results for a very well-known CB1 agonist, CP55940, from this work, which we published in January, and this reference given below, the work of Ayolua as well as Pankaj Pandey. You can see that in two of these structures, the molecule docks very well to the co-crystallized version of the molecule. That's showing that the glide docking does a very good job. That's the glide docking from software from Schrödinger. And then docking into the inactive versions of the protein, it does not do quite so well. And then we can then use the docking as part of a virtual screening funnel. For example, in this work, which we published in the journal Molecules in 2018, you can see we started with a group of 300,000 natural product-derived molecules, and then used a variety of filters, including docking, in order to find which molecules would be potential new hits for having actual experimental activity against the cannabinoid receptor as inverse agonists. So this project turned out very well to identify some structures which had suitable activity for further optimization. So we can also then, as a result of docking, study the predicted binding mode and see about different possible interactions, then design analogs which would be modified in order to have improved activity. So this project has worked out very well in that way. I'll show you some recent work we're getting ready to submit for publication, which a number of my research group members, as well as some collaborators, have been working on the COVID-19 spike protein and looking at some natural products, sugar molecules, to see if they're able to affect the ability of that protein to interact with, and to promote the life of the COVID-19 virus. And then we sometimes have also studied protein-protein interactions. Finally, here's one example of some work we've done in collaboration with our Pharmaceutics and Drug Delivery Department about the importance of cyclodextrins. And here you can see how we were able to predict about not only whether this cyclodextrin, given with the body representation, is able to help solubilize this progesterone drug, but also which end of the progesterone will be pointing in to the complex. So to summarize, docking can aid our understanding of how drugs work. We can learn about the binding modes. We can gain perhaps qualitative, possibly quantitative understanding of how the molecules have their effect. This can be helpful in identifying new leads which have the same mechanism of action. So one of the advantages these days, there are so many docking softwares available that docking is easy to do. However, I caution you that it's also easy to do wrong, incorrect, basically useless or unhelpful docking. So take care and check your methods carefully. So thanks again to all my collaborators and their research groups, as well as my own research group, for computer time, and thanks as well to you for your attention.
Thank you. Thanks a lot, Dr. Robert. Very nice talk on CAD, computer-aided designing. You are very nicely elaborate the CAD and its correlation with biological activity, and what kind of, here just I would like to ask one question only. What is the inference of different docking protocols which you have used in the CAD? Various kind of docking protocols.
Sorry, you said various kinds of docking protocols. What exactly is your question?
Yeah, the inference of different docking protocols.
Yes, as I mentioned in a couple of my comparative examples, the influence is very great. You can end up with complete nonsense if you're not careful. So the choice of docking protocol, we can't just say one size fits all or one approach works in all cases. You have to do detailed study.
Yeah. Thank you. May I request to Dr. Soren to ask few questions on docking? If you have any query,
Excuse me?
Yeah.
Excuse me?
Yeah.
I have a very generalized query in this. Like whatever computational tools we are using to find out some good molecules, lead molecules, but still the success story is very less. So whenever such molecules tested for biological activity, it is really not working so well as we predicted. So what can be the possible reason for that?
As I mentioned, sometimes it happens that although we sincerely try to use a particular computational method, it actually isn't the right method for the job. So one question is it possible ever to get good results from this type of approach? Well, yes, now the methods of docking are well used in the pharmaceutical industry as well as in academic labs around the world. And there have been a lot of successes where the result has been useful marketed drugs, which have, lead to the improved health of people around the world. So it is possible, but you just can't use docking by itself, and also you have to be cautious about which approach is needed. So for example, when we do docking, we also usually do now molecular dynamics simulations after the docking. We try to use various approaches to estimate more accurately what is the interaction energy between the small molecule and the protein. That's a great question, and it is an immense challenge, of course.
Yeah.
I have one question.
Thank you.
What physical-chemical properties we should monitor before docking any molecule in software or before docking with any protein site, what physicochemical properties or parameters we should monitor?
Yes, well, one thing to note is that basically docking programs can actually work without considering about the details of that nature. You know, you just need to know the molecular formula and the connectivity. So you can start with the so-called SMILE string, you know, which just illustrates all the atoms and their what's connected to what. So that's one of the nice things about it. But if you do want to consider about the physical chemical properties, such as the, you know, are there different regions of slightly positive or negative charge or things of that nature, then you can use that to perhaps choose what would be the most appropriate software. So just as an example, now we're doing a lot of research on glycoscience, and one of the key factors when sugar moieties are connected to each other, they have glycosidic bonds. And so for those glycosidic bonds, it turns out that the standard methods of docking usually don't do a good job of estimating what are the correct dihedral angles that would be appropriate because of the unique, you know, you could call it the unique properties of these rings containing oxygen, 5 and 6 membered carbon rings. So the result has been that special parameterization attempts have been made in order to include that information very accurately. And so you can use the glycam force field as an example to help with docking. And we use the software called Autodoc Vina Carb for that because it includes the appropriate parameterization.
So one more question. Is there any minimum number of molecules required before starting docking, or we can start with a single molecule also?
Yes, you can start with a single molecule. Sometimes it's nice to be able to compare, but oftentimes if you're just curious about one particular molecule, you can just go ahead and get started with that one.
Thank you, sir. Thank you very much.
Thank you.
Yeah, thank you, sir. Now, finally, today's weather connected with us, Shri Shyam Rathod, the chairman, Truba Group of institutions. So I would request to Shyam sir say a few words for our participants and participants and all audience. Shyam sir.
Thank you, Dr. Pawar. Am I audible?
Yeah.
Thank you, Dr. Pawar, for giving me an opportunity to speak in front of such esteemed guests. I welcome Dr. Robert, Dr. Suren Jain, and all the participants from different parts and all the faculty members. I think you must have gained a lot of good knowledge from the presentation given by Dr. Robert. I was also part of the presentation for a few minutes, but since it's not my say cup of tea, I could not understand the words, so I left the meeting. But that's a wonderful thing that you are getting all the experiences and the vast experience of Robert is you are going to get in one, say in an hour with so comfortable positions sitting at home, and you can get all the things. And thank you very much, Dr. Robert, for giving this. You're sharing your knowledge with all the Truba family and the students and the teachers. And I think it will be helpful for them for further growth if they want to go into research and develop their own CVs and all those things. Thanks once again, and have a wonderful evening. Thank you.
Thank you, sir.
Thanks very much.
I would request to Professor Dhote for vote of thanks. Professor Dhote?
Just a second.
Yeah.
Am I audible, sir?
Yeah.
On behalf of Truba Group of Institute, I would like to thank our eminent speaker, Dr. Robert Dodson, sir, who is joined us from the University of Mississippi, USA, for today's talk on molecular docking. I would like to thank our guest of honor, Dr. Deepti Jain ma'am, in RGPB Bhopal. I would like to thank respected Chairman Shri Shyam Rathore sir and all the DOG members for their guidance and support. I would like to thank Dr. Manish Manoria sir, Executive Director, Truba Group of Institutes, for his kind support and guidance. I would like to thank respected Suren Jain sir, Director, Truba Institute of Pharmacy, for his guidance and support. I would like to thank respected Dr. Rajesh Singh Pawar sir, Principal, Truba Institute of Pharmacy, and convener of this event, for the continuous support and efforts. I would like to thank the audience and participants from India and abroad for joining us in this web conclave, 11th web conclave. And I would like to thank all the faculty members, staff, and the members of Truba, Team Truba, for their continuous support and encouraging words for the event. Thank you very much. Thank you, sir.
Thank you. Thank you. Thank you, Robert, sir, for your valuable time.
Thank you very much. Thanks
Thank you.
to everybody.
I will discuss about you with respect to natural product, how we can work together with respect to natural product chemistry.
Great.
Okay, in future.
Thank you.
Okay, thank you. Thank you, sir. Thank you. Thank you, Suren, sir. Can we